Uh, I'm very shy, so that's why. If you support me, please ask something. So that's it. So anyway, it is my pleasure to have an opportunity like this. So recently we started discussion with our friend BioLasco how to provide our NOG mice to Taiwanese researchers. So that is why I came here. Unfortunately, like our principal investigator is coming today, so he is just arriving at the airport right now, but he is going to give a talk tomorrow at some conference, right? So, but listen, today I'm going to introduce our NOG mice and our second generation NOG mice, just like a summarize. So let me introduce our institute first. So our institute is Central Institute for Experimental Animals. So that is founded in 1952. It's almost 60 years ago, more than 60 years ago. So Tatsuji Nomura, who is an MD and PhD, so he, at this time in Japan there is no breeding company, and researchers are using very wild type mice, and that is not reproducible. So that's why he decided to make a research institute. Then we start developing animal, experimental animals. So right now we have 100 employees, but this is only for Central Institute for Experimental Animals. That means only for like a research purpose. We have another company, which is Korea Japan, and a breeding company. So we are researching about experimental animal in this institute and producing animal in breeding company. So and distribute to the all over. So here, right? So Korea Japan is our related company. So our purpose is just like here, to contribute to health and welfare of human and animals by overreaching basic research and clinical program. So that is very, very complicated. But mainly we try to develop animal model. The animal model should be reproducible, should be very close to human disease. That is our mission. So right now we are focusing on two models. One is NOG mice, which I will explain today. Another one is marmoset, which is a non-human primate. So today I will not talk about marmoset, but just briefly. So 2009, we are succeeded to generate transgenic marmoset, which is a world first success story to generate transgenic non-human primates. Actually, at that time, we developed GFP transgenic marmoset. After that, we are generating like Alzheimer's disease model, Parkinson's disease model, autism disease model, especially like a central nervous system. We are focused on central nervous system using marmoset. But mouse is more, wider purpose. That's why. So today I'm going to explain 3 or 4 research results. So before that, I want to explain what is an immunodeficient mice or what is an NOG mice. So as you may know, there are so many immunodeficient mice in the world. Maybe you are using noodle mice, you are using skid mice, or NOD skid mice, I don't know. So basically, noodle mice is a spontaneous immunodeficient model. It's more than 40 years old, right? We can find these mice, and a lot of researchers are still using noodle mice. So NOG mice is based on NOD background and also 2 mutations. It's KIT mutation and also IL-2 receptor gamma mutation. So that's why NOG mouse is IL-2 receptor gamma knockout and NOD skit based model. This is one of the most immunodeficient mice in the world. I would say one of, that means not only one. As you know, there is an NSG, which is developed by Jackson Laboratory in the United States. So a lot of people are asking, what is the difference between NOG mice and NSG mice? I answer always, just say it. But you are scientists, I have to explain detail. So the difference is how to knock out IL-2 receptor gamma. NOG is just, exon 6 and 7 truncated knockout model. Jackson Laboratories NSG mouse is, I don't remember, but exon 2 to 7 truncated model. That is the difference. But phenotype, okay, to be honest, we don't know because we never use NSG mice. But based on the paper, I think same. And of course, Jackson Laboratory guys, they never use NOG mice, so that's why they cannot say same. But we have, actually I have some small evidence because you may know Taconic, right? Taconic is US breeding company. So 3 years ago, anyway, Taconic is our licensee. So Taconic distribute NOG mice to US market and European market. So then 3 years ago, we recruit research director at Taconic, who used to work at Jackson Laboratory. So I think he is the only one person who use NOG mouse as well as NSG mouse alone. Then he said, just say. So phenotype is same. So that's why we are thinking that is same. So based on some manuscript, there is some small difference. I don't believe so. So some people do this research, some people do some other research. Of course, there are some differences, but basically, at least in terms of immunodeficiency, NOG and NSG is very, very close, and I would say same. So that's why NOG and NSG is a very good model as a, we say, I don't know. Some people say super immunodeficient mice, or we say severely immunodeficient mice. So we can say one more super immunodeficient mouse, which is, we say BRG mouse. This is a B6 background and IL-2 receptor and LAG2 double knockout mouse. So actually we can provide this mouse to in Japanese market. But we basically recommend to use NOG mice. I will explain later why. But in terms of immunodeficiency, this mice is also same, not same, we cannot say, similar to NOG or NSG mice. But of course, we are doing research even now based on NOG mice. So today I will explain second generation of NOG mice. That means today I will show 3 lines, but behind the 3 lines we already have 80 or 70 or 80 lines based on NOG mice. That's why please use NOG mice for your future research. So that is why we strongly recommended NOG mice instead of these mice. So anyway, so as I explained, NOG mice is NOD inbred strain and SKIT IL-2 receptor gamma mutation, right? So from NOD inbred strain, so we can see reduced NK and complement and macrophage activity, they are reduced. And from SKIT mutation, effect of T and B cells. And from from IL-2 receptor gamma mutation, we can see the effect of NK and dendritic cells function. So NOG mouse has all of them. That means of course, there is still macrophage activity, but a lot of cells are not existing in mice. So that's why, as far as I know, this is very, very good model for humanized or PDX or anything. But again, this phenotype is just similar or same with NSG mice. So I will explain 2 or 3 slides. I will explain about NOG mice, compared to NOD skip mice. So I think some of you are using NOD skip mice, right? This is the difference of tumor engrafting, right? Yeah. So we picked up pancreatic cell lines, and of course human pancreatic cell lines, tumor cell lines. So this is a cell dose which we injected, right? From a small amount of cell to large amount of cells. Of course, we picked up some difficult tumor lines which cannot be engrafted in NOD skin. You can see these 3 lines. So NOD skin mice cannot accept this tumor, even like a big amount of cell juice, like this. But NOG mice, NOG can accept this, not only this, but also a lot of cell lines. Even smaller amount of cells, so some of mice can be accepted this tumor. The difference is just immunodeficiency. As you can see, NOD skin is just this and this, right? So, but NOG has also IL-2 receptor gamma mutation. That means there is no NK cells and no dendritic cells. So that is why immunodeficiency is much more, I don't know how to better or worse, but it's severe compared to NOD skin. That's why there is a difference. But again, NOG and NSG show same result. Not only NOG, but also NSG can also show this kind. So this is the same tumor. One more good thing using NOG as a PDX model mouse. So there are, you can see different tumor lines, right? Then you can see different, I don't know how to say, different phenotype of tumors. They are different. They are different looking, right? So that means NOG mice has no immunology, immune system. That's why the tumor can be original. So there is no difference from the original tumor. So you can see the difference. So it's hard to say in English. We can keep original phenotype of tumor cell line in NOG mice. That is what I want to say. So they are different tumor lines, and you can see difference. So we can keep this difference in the NOG mice. That's why some people are using NOG mice for PDX model. Even NOG mice is expensive compared to NODs. So this slide is just explain NOG mice. So NOG is very, very good mouse model for PDX model, right? Because of its severe immunodeficiency. But one more good thing using NOG mice is like a humanized mice. We can generate humanized mice using NOG mice. I will explain slide. So here is a procedure how to generate NOG mice, but it may be too small. But not that difficult, it's easy. Just X-ray irradiation. After that, we inject human hematopoietic stem cells. Then after 30 amount of time, we can see some of human immune system in NOG mice. So far, we can say NOG mice can constitute of human immune system from like these cells. But unfortunately, we cannot see developed erythrocyte or megakaryocyte. So now we try to generate these cells also, but so far we cannot detect developed erythrocyte in normal. I mean human erythrocyte. But I will explain detail later, but normalized is very, very well developed T cell and B cell. So that's why so far, if you are very interested in human T cell or human B cell, normalized is very, very good model to generate human T cell and B cell. This phenotype is also similar to NS mice. Without like this, again, I'm compared with NOD skin and NOG mice. After engraftment of human hematopoiesis stem cells, so this is peripheral blood, and we can compare human CD45. So as you can see, there is big difference between NOG mice and NOD skin mice. Actually, NOD skin mice, you cannot generate humanized mice in terms of hematopoiesis stem cells, because NOD skin has still NK cells or some immune system. This is immune system reject human hematopoiesis stem, so that's why. So this is in peripheral blood from 8 weeks to 20 weeks after human hematopoietic stem cell injection. You cannot see any human CD45 in peripheral blood as well as bone marrow and spleen. So only NOG mice can detect human CD45. So that difference is, again, just simply the difference of immunodeficiency. So NOG is much, much better, I mean, severe immunodeficiency compared to NOD skin mice. That's why we can generate humanized mice. In this case, humanized means human immune system response to mice. So far, as I showed, like a nog mouse is very good for PDX model as well as humanized mouse model. There are a lot of researchers all over the world is using NOG mice for infection disease model, like this, the cancer model, like a PDX model, and also other models like a GVHD, everything. So recently, you may know, now in Japan, like IPS is very, very hot, and Japanese government decided to use a lot of money for IPS research. And nog mouse is officially approved to be a, I don't know, say final preclinical study for IPS study. So before clinical study, we have to use nog mouse. So like a IPS derived some kind of cell should be engrafted in nog mice. Then we have to check this cell will be a cancer or not cancer. If it will be a cancer, we cannot put this cell into the human. But if there is no cancer in the nog mice, we can use these cells for clinical study. That is Japanese law, right? But actually NSG, we can use it. So not only NOG mice. But anyway, so that's why now NOG is very, very wide application. But still, today I want to explain something. Still there is a limitation of nog mice. That's why we keep research. So we call second generation. This is how to generate humanized mice, right? So after 2.5 gray irradiation, we inject human CD34, which is hematopoietic stem cell, into nog mice. Then we can get human immune system in nog mice. That is humanized nog mice. Okay, so the data is, this is human peripheral blood. This is humanized nog mouse peripheral blood. So regarding, about lymphoid cells, it's okay. Even humanized nog mice, we can see a lot of human lymphoid cells. That is no problem. But the problem is myeloid cells, like this. human peripheral blood, you can see a lot of myeloid cells, right? But humanized log mice, yes, we can detect, but the number is very, very small. So in terms of paper, we can say, oh, log mice can develop myeloid cells. So that's why we check the positive. But actually, the number is very small. Like, for example, like a pharmaceutical company, their researchers are not satisfied with this number because no, we cannot do research using these mice. So that's why we try to generate another rounds based on the mice. So it's okay. This is our, sorry, this is very busy. It's right. But the strategy to generate second energy mice is sometimes we put human genes into log mice. Sometimes we delete mouse genes from log mice. Sometimes we put virus genes into log mice. So we modify log mice, and then we try to generate better. That is our big picture. So today I'm going to, I'm going to 3 examples of our second generation log mice. This is one first. So as I explained, log mice but log mice is less efficient to generate myeloid cells, right? So this is our hypothesis. So of course, hematopoietic stem cells came from human, but in log mice, these cytokines, of course, came from mice, right? For example, like a lymphoid, it's okay. Human hematopoietic stem cell can, I don't know how to say, good affinity with mouse these cytokines. That's why human hematopoietic stem cells can be developed into lymphoid cells in log mice. On the other hand, these cytokines, mouse cytokines are not good, not crossed with human hematopoietic cells. That's why this direction is not working well. So that is our hypothesis. So that's why in this case, we induce human IL-3 and human GM-CSF, 2 human cytokines into log mice. Okay. Actually, this is the same protocol, but So this red line is our second generation, which is IL-3 and human IL-3, human GM-CSF transgenic log mice. Non-TG means conventional log mice, like old log mice. So as you can see, human CD45 is much better than regular log mice. But we already published 2013. So at that time, we can, it's kind of chimeric ratio, humanized chimeric ratio. Like a regular log mice is 20 or 25%, but this new line is more than 40%. So after that, this is CIA policy. After that, we try to establish very good lines. Then actually, we can get over 80% chimeric ratio lines, but this line does not work well. This line shows very severe anemia, and a lot of mice is going to die. So we are trying to, what is the problem? But we are thinking, these lines can generate a lot of human macrophages, and human macrophages attack mouse red blood cell. That's why this line induces mouse anemia. That is a problem. So that's why we try to change slightly. For example, this irradiation will be decreased 1.0 grade, and this cell also, we don't need this kind of cell. Now we are using 1 times 10 force. Then the final chimeric ratio will be like 60%. Then anemia is very, very high, so we can use this. So anyway, this line, of course, there is a small problem still, but in terms of in terms of humanization, this line is much, much better than all of them. So basically, we try to generate myeloid cells using this line, right? IL-6 This is all normal mice. So we can get a lot of myeloid cells compared to normal mice. But not only myeloid cells, but also monocyte. Even T cell, we can get much more T cell compared to normal mice. That's why we are thinking that these mice, new second generation of mice is much better than the old mice in terms of humanization. So this is like a time course after injection, after a human hematopoiesis stem cell injection. You can see a lot of granulocyte. Of course, myeloid, it's much better than the old mice. And granulocyte also, monocyte also. Even T cell, we can get more. But B cell is, old mice is better than IL-6 So not only myeloid cells, but also a lot of immune cells are generated in IL-6 and CSF normal mice, much better than regular, conventional mice. So not only peripheral blood, but also in spleen, we can actually this mice has bigger spleen compared to normal mice. Actually, normal mice has very small spleen. But anyway, we can get, so in this slide, you can see the difference. Cell number, it's huge difference because of the size, of course. But you can get a lot of big amount of cells instead, compared to normal mice. Yeah, not only I cannot explain everything, but a lot of cells are ILC and GFCS normal mice is much better than regular, conventional mice. So in near future, we try to change from normal mice to IL-3 GM-CSF normal mice because this new line is much, much better than conventional mice. Actually, that is a difference from NSG mice. So we are preparing second generation. So NOG is same with NSG, but this second generation is much, much different from not only NOG, but also NSG. Not only FACS data, but also you can see immune staining. So you can see eosinophilia, of course, human eosinophilia and basophil. Unfortunately, so I want to show neutrophil here, but I can see some neutrophil, human neutrophil in this side, but like a CIA scientist is very, very, very strict about this kind of stuff. Because, CIA is, of course, a research institute, but our mission or our goal is not publishing the paper. Our goal is develop animal model and provide this animal model to researchers. The researcher will use this animal model. That is our purpose or our goal. So if I show you your champion data, so you might be misleading. That is CIA's researchers very, very strict about this. So that's why I can show you like a neutrophil data here, but my coworker who is coming to the airport right now, he said, no, you cannot show this thing because this is still not developed well. So that's why I have to wait that picture. But still, okay. So I can say, we can see human eosinophil and basophil. Neutrophil is still very small, not enough developed. This is FC receptors. You can see the much difference compared to regular mice and the IL-6 not only peripheral but also these a lot of organs. So that means immune cells are working well or functional. So finally, I will show some in vivo data. So I want to say in vivo, but it's slightly artificial in vivo because even normal mice still we cannot succeed to generate human antibody in no mice. So that is very, very difficult. But we are trying right now. But anyway, in this case, antibody and allergen is injected from outside, right? So at the same time, we can put it like a color, a blue color. So if there is an allergic reaction, you can see blue color in the skin. So that is very, it's kind of old experiment. So it's easy to see, right? So conventional mice, yeah, we can see small, blue color, but this new line, you can see a lot of blue color. That means the allergic reaction is happen in this new generation because there are so many immune systems and immune cells in those mice. So we already published this data, and we already started to provide IL-3 GM-CSF mice to all over the world. So far, as far as I know, a lot of Japanese pharmaceutical companies and US pharmaceutical companies, European pharmaceutical companies are really interested in this line, and some of them are already using this line. So we can provide this line maybe to you, but in this, if you are interested in this mice, please ask Biographical. We can arrange how to provide. So this is one example. So I will explain two more examples. Another one is IL-2 receptor transgenic mice. That is also same strategy to generate second. Before this line, so we introduce IL-3 and GM-CSF, two human cytokines into no mice, right? In this case, human IL-2 cytokines are induced in no mice to produce human NK cells. Of course, regular no mice, conventional no mice also can generate NK cells, but the number is very small. So that's why we try to generate this way. This year we published the data. If you want to, you can get a lot of data in this publication. But anyway, so black box is conventional, right? White box is IL-2 transgenic. So after 6 weeks of human hematopoietic stem cell injection, so conventional mice is mainly B cells. Human B cells are major cells. But human IL-2 transgenic mice, as you can see, a lot of NK cells. But small amount of B cells. But so many NK cells are produced in IL-2 transgenic. That is what we want to generate. Of course, not only peripheral blood, but also bone marrow, spleen, peripheral blood, everywhere. So, human cellular cancer. So, that's complicated. So, black box is NK cell, right? So, IL-2 transgenic mouse has a lot of NK cell in peripheral blood, spleen, and lung, and liver. Bone marrow is very small. But conventional there is no, actually there are some NK cells, but it is difficult to detect, right? So that's why these new lines can produce a lot of human NK cells compared to regular mice. So not only facts data, but also we have to make sure these NK cells are functional or not, right? So we checked the expression of nuclear receptor. So this is IL-2 transgenic mice, humanized IL-2 transgenic mice. This is human peripheral blood NK cells. So receptor is similar, right? So that means our humanized mouse NK cell is also functional, not only NCR but also this family too. Also we checked KIR family, KIR family. So in this mouse, there is an expression. We can detect the expression KIR. I don't know if it's English or not. KIR family. Maybe KIR family. Right? Yeah. And also, we checked granzyme A and parvorin, which are proteins released from NK cells. So this is peripheral blood of human. Human peripheral blood NK cells, right? So granzyme A and parvorin are released from NK cells. So this release will be observed in our humanized IL-2 transgenic mice. That means, like an NK cell which is developed in IL-2 transgenic mice also function. Then I will show two more data, in vitro and in vivo study. This is in vitro study. That's slightly confused or easy. I will explain this at least. So this is in vitro data using K562 tumor lines. So we checked cytotoxicity. These tumor lines are very, very sensitive against NK cells, NK cytotoxicity. This open circular is NK activity came from human peripheral blood. Of course, human peripheral blood NK cells has very strong cytotoxicity activity against K562. You can see strong cytotoxicity activity. From IL-2 transgenic mice, we can see some relatively weak cytotoxicity assay compared to human peripheral blood. Right? But from this point, we add human cytokines like this. This bar means this one. Right? So if we can add human IL-2, that this cytotoxicity activity is going up to close to 50%. If we add IL-12, it's almost IL-15, we can get more than 60% cytotoxicity. So the combination is also good. So that means our NK cells came from human IL-2 transgenic mice is relatively weak compared to K562. But if we can add some cytokines, the activity is going up. So I don't know why this is going to happen. But basically, this mouse line is IL-2 is constitutively expressed in the nog mice. The concentration is actually 10 times more than like our body. That means this NK cell is slightly dependent on like a cytokines. That is our speculation, but we are not sure. So anyway, this is in vitro data. Of course, we need in vivo data, right? So in this case, we are going to show two different kinds of tumor, like a K562. I already explained in vitro study. This is in vivo study, right? So humanized IL-2 transgenic nog mice, K562 cannot grow because this line has tons of NK cells, as you can see before. That's why K562 cannot grow in this nog mice. That means NK cell can active against K562 based on this data is the same with in vitro. So compared to IL-2 transgenic mice, these two are conventional mice. So that means the difference is 6 weeks after hematopoietic stem cell engraftment or 12 weeks after. The difference is just time. Actually, this is 6 weeks. So this means the difference is 6 weeks after means almost all immune cell is a B cell. 20 weeks after transfer is like a T cell is a measure. That is the difference. Even B cell or T cell, they cannot stop the growth of K562 tumor. So that is why we are thinking this suppress of tumor growth is depend on NK cells. So anyway, this is the data published this year, but that is also like a CIEA's policy. These lines means individual mouse, not average. So sometimes like a CIEA researcher says, average is also misleading. So, we have to show the diversity, right? So that is why always they are doing this. Each individual shows, right? There is some difference, of course. But that is very clear, right? You cannot see. Yes. Then this is cytotoxicity activity in vivo study and ADCC activity. That is also very interested in, pharmaceutical companies are interested in ADCC activity because they try to make a drug. That is also complicated, right? So in this case, we are using L428 Hodgkin lymphoma and CCR4 antibody. This is kind of very famous combination. This is IL-2 transgenic pneumovirus. And these two are humanized IL-2 transgenic pneumovirus. The difference is no NK cells or a lot of NK cells. And the left and right, the difference is without antibody or with antibody. Then this is tumor growth. Again, the one line is one individual. For example, like this graph shows no NK cells, no antibody. That means we cannot stop the growth of Hodgkin lymphoma. Four animals. In 4 animals, we can see the growth of tumor. Right? Okay, in this case, no NK cells, but with antibody. But still, we cannot stop the growth of tumor. Yes. Okay, this case, with NK cells, but without antibody. In this case, still we cannot stop the growth of tumor because L428 Hodgkin lymphoma is less sensitive to NK cell cytotoxicity. For example, like K562 is very sensitive. That's why only NK cells can stop the growth of tumor. But this tumor cannot. We cannot stop the growth of tumor. But NK cell with antibody, we can stop the growth of tumor. This is ADCC activity, right? We can show ADCC activity in mice. That is our result. Basically, this IL-2 transgenic mouse was developed by NK cells. That means we want to make a new line for ADCC activity. That is why we already published this line, and we already start provide this line to Japanese pharmaceutical companies. So finally, these two lines are human cytokine, right, and to reconstitute human immune system in dog mice. The last one is more slightly different. This is humanized liver mice. So that means the mice has human liver. That's weird. Right? So it takes time to explain that. Human liver is very, very important organ for drug discovery. Of course, we need to check, drug metabolism or drug response, toxicity. The liver is very, very important. Right now, a lot of pharmaceutical companies or even like researchers, same as you, try to check this with, for example, a transgenic mouse for in vivo study or primary culture, human hepatocyte in vitro study. But okay, most of them are good, but we generate humanized liver mice which has human hepatocyte in mice. then you can do in vivo study. That is very, very good, right? So basically, you can use this line for drug metabolism, infection disease such as HCV and HBV or malaria. Actually, we published malaria disease. And toxicology, of course, regenerative medicine and other disease model like cancer, liver cancer. But this line is very, very, it's hard to generate, actually. So to generate this mice, we think the three important things. First of all, the mouse should be super immunodeficient mice. Otherwise, of course, mouse can reject human hepatocyte. Of course. So that's why, of course, our nog mice is very, very good model or based on this one. And also, we have to show, we have to induce chronic liver, mouse liver injury. And in this case, we are using TK, which is chimaging kinase from vaccine and virus, and GCV system are algorithms later. And also, we have to get donor cells. That means human hepatocyte. This human hepatocyte is also important to recover this mouse injury. Okay. So basically, this model is like a TK. Okay, I will explain how to generate this model. So TK is a chimaging kinase, which is that's hard to explain, hard to pronounce. Suicidal genes, right? Suicide, yeah, that's killed by themselves. So this chimaging kinase is expressed in only liver. So that is our design. Okay. Only liver, right? You can see only liver, no other organs. Only liver, this chimaging kinase is expressed. Then, when we put gancyclovir, GCV, the switch is turned on. And this, chimaging kinase is going to start to express. Then we can see liver injury. So, of course, mouse liver injury. And certain amount of injury we can detect. We inject human liver cells from the spleen. Then human hepatocyte can grow, but mouse liver is going to disappear. Then finally, mouse liver is replaced by human liver cells. So this is the data, of course, and a lot of human CYP genes are expressed in humanized liver mouse. Then, as you can see, this is a kind of example. So this is, of course, humanized liver, mouse liver, stained by human antibody. That means, for example, like this spot is not stained, right? That means still there is some mouse liver, but the other part is very good, humanized, means human cells. So we checked this staining and checked chimeric ratio. Then actually this chimeric ratio is very, very parallel with plasma human albumin. So that's why we can check human plasma human albumin, then we can predict the chimeric ratio. That's very good data. So otherwise, we cannot speculate how much chimeric ratio. So that's why we use this. So actually, right now, our humanized liver is almost, this is also provided by us to the all over the world. For example, like this. This is conventional nog mice. Of course, this conventional nog mice has no human liver. That's why stained by HLA, there is no, of course, there is no stain. But our humanized liver, this is two humanized liver. For example, like this, it's almost all liver are composed of human cells. This line, you can see here, this is mouse, and also here, This is also mouse. So if you can see enlarged, here is a human and mouse spot. But in this case, more than 95% is human. Even this case, more than 90% are humans. Yeah, that is our new model. Not only like HLA, but also a lot of enzymes we check. But we can see a lot of. I will explain detail later, especially this one and this one. Oh, that's a complicated story. You are not interested in liver, maybe? Anyway, so liver is, of course, as you can see, our humanized liver mouse has human liver cells in liver. But of course, liver has some role. Not all cells has same role, right? For example, like this. This is central valve. This is central vein, and this peripheral vein. There is a difference, for example, like a metabolite or a nutrition oxygen. For example, like a metabolite is central veins much concentrated, but not peripheral vein or something like this. There is a difference, not only this metabolite, but also gene expression. So that's why I showed two examples. CYP3A4, which is one of the CYP enzyme. This is human liver, right? If we can stain CYP3A4 in human liver, this enzyme is expressed only around the central vein, not peripheral vein. Our humanized mice also shows similar pattern or phenotype or expression level. So the expression is located on central, not on peripheral. So that is why it's similar. Gene expression is also similar. And this is glutamine synthesis. Actually, this antibody is react with human and mouse antibody. But anyway, so this is also located around central vein, not peripheral vein. That is also same with human expression. So that is why our humanized liver mouse shows high chimeric ratio human cells in liver, and also shows, I would say, function. Not only gene expression, but also I will explain later. I will show. Oh, sorry, some of them are Japanese. I cannot change. This is very famous, thalidomide, right? This is, you can see this kind of baby using thalidomide. The difference, sorry, my PI gave me this PDF file. It's not PowerPoint, that's why I cannot change Japanese. Anyway, so that is my excuse. So in human and in mouse, there is a small difference. You can see the difference. In liver, thalidomide is going to add OH at this location in mice. But in human, OH is added this point. That is the difference between mouse and human. But in mouse, this compound actually less reactive to the compound. That is why it's safe, no toxic. But in human, this part is slightly active point. That's why we can get more side product like this and like this. These compounds are actually really toxic. That's why we had a very serious accident using thalidomide. So the answer is our humanized liver mouse can detect this kind of side product like this. So that, of course, our humanized liver mouse has human liver. That's why the human liver can produce this side. Also, that is very complicated slide, but you cannot. Actually, it is hard to see. But anyway, this is a kind of drug which shows different metabolite in mouse and in human. So it's okay. I just say this is human-specific product. We can see only humanized mouse. We can see the human-specific product, but there is no mouse-specific product like this. So this is so in T10 of mice, not humanized mouse, we can see mouse-specific product, but not human-specific product. That is very clear, right? Our humanized liver mouse shows human metabolite. That's why we can offer this mouse for drug discovery and safety study or toxicology. Oh, okay. That's fine. So I'm going to explain it. So as you might know, like that's really difficult to explain. Anyway, so difference of molecular seeding between human and mice. Right? So for example, like the molecular weight more than 500 in human is going to bio, but less than 500 is going to urine. But the molecular cut is different between human and mice. For example, like mice, less than 300 molecular weight is going to urine, but more than 500 is going to bio. So we chose this molecule because this molecule is 390. That means between this. So if our humanized mice liver is going to show same result with human, this compound is going to urine, not bio. So actually we can see that. That's complicated, but this is the result. So that means our humanized liver mouse also shows same molecular seeding with human, not mouse. That means that this kind of stuff is very important for like a metabolism study for drug discovery. That's why we put this data to. Okay, sorry. It's almost time. So, and not only like a metabolism study or toxicology study, but also a lot of infection models we are using this TKM humanized liver. This is this is HP virus, right? Oh, this year we published Nature Communications using this model for malaria study. So not only like drug discovery, but also like infection model. This model is very, very useful. Today, that's fine. Today I showed 3 examples, right? But actually CIA has, I don't know exactly, but more than 70 lines, maybe 80 second generation. Today I showed like a myeloid cells, NK cells, human liver cells. So if you are interested in some human cells, we can produce in dog mice. That is my essence. So that is why please use dog mice as a first step. Then if you want to, I don't know how to say, study more, you can have a choice to use second generation. So if you are interested in more, so I can explain. So anyway, that is time. Thank you. Thank you for your attention.